Hello everyone, my name is Luca and I'm a second-year biomedical engineering student at Glasgow University, and I want to talk about the research project I took part into last summer on cancer genomics. The main aim of the project was to identify genetic mutations which might result in cancer. So first of all, what is cancer? How do we find out? But most importantly, why is cancer related to DNA? So we know that cancers have been found in remains from ancient Egypt, and the writings from around the same time confirm that cancer was actually documented by these people. The picture you see on the top, if the pointer works, shows metastatic prostate cancer in the lumbar region of the spine of a 2,150-year-old mummy. But the first to actually describe cancer were the Greek. So Hippocrates is the guy that you see there, and he describes cancer. That's why all the names that are related to cancer somehow, such as carcino, carcinoma, and even onco, from which we get oncology, all those names come from Greek, and onco in particular means bulk. So all the scientists from all over the following century started investigating the causes of cancer, but in the 17th and 18th century, people still believed that cancer was contagious, and we now know that that's not the case at all. So it wasn't really until the beginning of the 20th century that, thanks to the work of scientists such as Watson and Crick, that we began to understand the structure of the DNA, how DNA works, but most importantly, why is DNA related to cancer? So let's give you some numbers. So we know that 1 in 3 individuals will develop cancer at a certain point in their lives. In 2012, 14.1 million cancers have been diagnosed worldwide, and it turned out to be slightly more common in men than in women. The most common location for cancer, accounting for 13% of the cases, was lung cancer. However, the most common cancer for women is breast cancer, and for men is prostate cancer, as you can see in the pictures. Well, although cancer can affect pretty much anyone at any age, we know that it is predominantly a disease of old age. This is why every day we are exposed to some factors such as pollution, chemicals, smoke, radiation from the sun, or even some viruses. And all these factors can damage our DNA. And wait for the video, right? And yeah, if the genes which are damaged were involved in some vital processes such as cell survival, proliferation, or even motility, that could be a step on the pathway to cancer, as shown by the red spots. What happens is that we need 6 hits, 6 events, before getting cancer. So now we live long enough to accumulate all the damage which is needed to get cancer. That's why the graph shows that the total number of cancer cases increases as we get older. And the reason why cancer genomics has become increasingly important is because now we live, nowadays it's quite cheap and fast sequencing the entire DNA. What we want to do is comparing the DNA of a normal cell with the DNA of a cancer cell and see if there are any differences. I said sequencing. So what is sequencing? Sequencing means reading the DNA, because we know that the DNA is made of 4 main components, which are called bases or nucleotides, and these components are A, C, G, and T. So what we want to know is reading the DNA, reading the sequence of all these 4 letters, and see again if there are any differences. Usually we sequence the genome, which is the sequence of these letters, but sometimes this could be quite long, so we're interested in just the bits that will eventually become RNA and will eventually give proteins. And when we do that, we don't sequence the genome, we sequence something that is called transcriptome. So during the summer, I mainly dealt with the genome, and the samples that were taken from people having colorectal cancer, while the analysis on the transcriptome was carried out on people having thyroid cancer. And they also evaluated the toxicity of potential anti-cancer cell drugs on different cancer cell lines, such as breast cancer, cervical cancer, prostate cancer, and so on. There are lots of ways to do it. The way we did it was using a technology which was partially developed at Glasgow Uni, and nowadays it is quite common all over the labs. So the way it works, we start off with a double-stranded DNA, we break it down into millions of fragments, and each of these fragments will then attach to a bead. it will be then copied over and over again till it covers the entire bead. And this process happens simultaneously for every bead. Then each of those beads will go to a well, and wells make up the chip. Then the chip is washed over with a different nucleotides every 15 seconds. Yes, and whenever a nucleotide is incorporated, what happens is that a hydrogen ion is released. Hydrogen ion released are chemical informations, and then what we want to do is converting this chemical information into digital information as voltage differences. Again, DNA is made of 4 bases, 4 components, the letters that you can see there. And we know that there's a specific voltage difference, specific voltage change, which is associated with all of those components. What happens is that DNA is made of 2 main structures, and those are linked by some links that can happen just between some of those bases. C's go with G's, and A's go with T's. This is the example of cytosine, which is the C, and whenever it binds to the complementary base, which is the G, hydrogen ion is released, voltage change is recorded, and therefore the base is called. If there's no complementary base, nothing happens. On the other hand, if there are 2 complementary bases which are adjacent, as in this case, there are 2 hydrogen ion released, the voltage doubles, and that is recorded by the machine. So the machine itself is quite clever. It can understand when there are different nucleotides in a row. It can be up to 8, then it kind of becomes messed up. And the good thing about this machine is it's that it takes just a small fraction of the time it used to take with previous methods, just 3 hours compared to 37 hours that it used to take with, again, previous methods. So why is this all useful? This has a huge potential for diagnosis, prognosis, and treatment. What we want to do, again, is comparing the 2 sequences of the DNA, and we want to spot any difference. Differences are called mutations, and there are so many mutations. I don't want to bother you with those right now. But when it comes to medical research, we're interested in 2 main types of mutations. So passenger mutations, which do not really cause cancer. They're just there because of the genomic instability of a cancer cell. On the other hand, we've got driver mutations, which are actually the ones which cause cancer. And therefore, they're the potential targets for cancer therapies. So what we want to do is sequencing the patient's cancer, classify it into a particular subtype, and that's an example. That is breast cancer. I know the picture is quite blurred, but what I'm interested in is those 4 subtypes of cancer associated with a different color and with different mutations that cause the cancer. Then we associate each of these subtypes with a relatively good or bad prognosis, and we tailor the treatment depending on the subtype. So what we want to do is treating the patient with a specific subtypes, with a specific treatment, which depends on the subtype of the cancer, which depends on the mutations of the cancer itself. Of course, this can have this can be extremely beneficial for the patient, as what we want to do is reducing the side effects of the treatment and give the patients a better chance of success than if the treatment was used for everyone. So this is what we call personalized medicine, which has, again, a huge potential, but this is why lots of research is still to be done on cancer genomics. And I just want to thank you for your attention, and feel free to ask any question if you have any.
You mentioned 6 stages towards cancer.
Oh, yeah.
What are these?
Right. In order to answer this question, we need to understand what a cancer cell is. That goes into depth of cancer biology. So what a cancer cell does is duplicating quite fast. So usually cells receive some signals from the other cells, which could be anti-growth signals or growth signals. So a cancer cell ignores all the anti-growth signals because cancer cells give some kind of bulk, so they should ignore all the anti-growth signals from the other cells. That's one. The other one is that they should be growth signal sufficient because there they wouldn't get any growth signals from other cells. Plus, they should avoid apoptosis. Apoptosis, cell suicide. So whenever cells duplicate and they make mistakes while duplicating, I mean DNA mistakes, there are some signals from the other cell, from the other cells, telling the cell to die, and cancer cells should say, no, I don't want to die. So they just ignore these kind of signals. Plus, they need lots of nutrients, so they should be able to construct their own blood vessel, and so this is what is called hemogenesis. And they should give metastasis, which means that the cancer has to spread, and then they should be able to proliferate quite limitless in order to be cancer. These are the 6 hallmarks of cancer. And there are some current research carried out by an Italian immunologist, and he says that if there are any infections in the tissues, all these 6 hallmarks can be quite, can happen more likely. Yes.
Yep.
Hi, I have a question regarding the methods that you use to measure the voltage difference between the 4 main building blocks of DNA. How long would the process take, and how long would the process take to get the voltage difference between the DNA sequencing, and how would you categorize the level of cancer in the DNA if it's present?
Right. Those are 2 questions which are not related to each other, right? Right. So, yeah, the DNA, when it forms, again, there are those 2 structures that pair up, and depending on the, in nature, this is done by an enzyme which is called polymerase, and whenever that happens, that's a chemical reaction. One of those products of chemical reaction is hydrogen. Hydrogen has a positive charge, and charges, we can associate charges with electrical potentials. And depending on the bases that pair up, there's a different amount of positive charges, and that corresponds to a different voltage, and we record it. Then you asked how long it takes to sequence a DNA, 3 hours. And then a third question was again?
How you categorize the severity of the cancer if it's present?
The severity of the cancer, you said?
Yes.
Right. Where's the point? There are, all right, those are the mutations, right? And there are clinical trials. Well, that's just, something that has to do with statistics. So, there are some mutations which are associated with specific types of cancer, while some others some genes are mutated in different types of cancer. So then we know that if someone has got a particular mutation, let's say the first one, and there are like 7 in 10 individuals who died after 3 years from the diagnosis, we can say how dangerous, let's say, a mutation can be when we do some kind of statistical analysis, Okay.
Any more? Yeah, there you go.
So I know absolutely very little about this side of science and science in general. It's probably a really basic question. I was wondering, you mentioned at the start how obviously with age more people get cancer.
Mm-hmm.
So then what happens when really young children get cancer? Is that just bad luck, or?
It's bad luck, but you can actually predict it because there are some kind of genetic trees. So you can get your mutations from your parents, and if you get one from your mom and one from your dad and the other one from your grandpa and the last one from your grandma, yes, you're quite lucky. There are cancers which are associated with a young age, but that's because, for example, retinoblastoma is a cancer in the eye, and that's associated with young cases, and that's because, well, the reason for that is because the gene which is involved doesn't let you live long enough to be 75, for example. I'm saying 75 because a third of the cases of cancer are related to people over the age of 75, but yeah, it's like genes are like the answer to your question. Any more questions?
Yeah, on you.
Yeah, wouldn't the personalized treatment be really, really expensive?
I think so, yeah. There are, well, even this process, well, it's relatively cheap, but it's quite expensive itself. But yeah, for example, that machine costs around $400,000, and for every run, you need to, well, it costs you $2,000 again, and even the personalized medicine, the personalized treatment would be crazy expensive. I totally agree. The fact is that 1 in 3 individuals will develop cancer, so I think they need to work out, you know, if it's worthy or not to do it. But cancer is quite it's quite a problem nowadays. So I don't know what they're going to do. Like when you do this kind of research, you go from point A to point B. I haven't done anything which is related to pharmacology, for example. So coming up with the idea of something that can really cure cancer. So I don't know how expensive that could be, but if you say I think it's pretty expensive, I totally agree with you. Yeah.
Yep.
I have a question about the personalized medicine. We can use it only in cancers that are caused by genetic mutations, but also by cancers that are caused by viruses like HPV.
So the papilloma virus, well, I think, well, when you get papilloma virus, for what I know, like I'm an engineer again, when you get papilloma virus, it doesn't really affect your DNA, for what I know, or well, you get it from, yeah, well, it's not from your genes, you know. So I don't think that can actually help, or we can see if the body responds to the virus in a different way depending on the mutations. That could be a good answer, I guess. But if it doesn't, I don't see the point in acting on the genes if the thing that you have hasn't been caused by the genes themselves. That's what I know, sorry. Okay, I don't know.
Hello? I was actually just going to jump on the same question and ask if you would be able to sort of determine whether there are pre-existing mutations that would make you more likely to develop cancer following being exposed to a virus, in which case would it still be useful for that same analysis?
Yes.
Yeah.
Yeah, absolutely, yeah.
But in that case, would you need to run that prior to, or as in if you're exposed to the virus, would you need to essentially get yourself tested for that? That would be a possible application?
Yes, of course. Now, like, while you're talking, I'm just realizing that actually, I think in the UK, 70 or 80% of cervical cancer come from the papilloma virus. I don't really know the cause for that, to be honest, like if it's genetic or not, but we should work on that. Well, for your question, yes, I think, yeah, if it's something that eventually will then be related to genes, we could do, well, we can work, again, tailoring the treatment to what has really caused cancer, but yeah.
So can we once again please say thank you very much to Luca?